Good morning. My name is Joy Concepcion. I am the Business Development Director for QPS, covering the Pacific Northwest. QPS is a global service provider to the pharmaceutical, biotechnology, and generic industries, and is strategically located with sites in North America, Europe, and Asia. We have over 1,200 global employees and counting. Today, I will focus more on our bioanalysis capabilities. QPS offers technical depth and breadth plus seamless integrated service from drug discovery to regulatory submission, providing sponsors the capability to develop their lead candidates all in one place. QPS is known for being agile, nimble, and fast, particularly during the current COVID pandemic, where companies had to postpone clinical trials and pivot to early development and preclinical studies that they could no longer support in their labs. Linear integration saves time, resources, and maintains the expertise within your program. QPS has a global bioanalysis team of over 360 specialists to support small molecules, biotherapeutics, and gene therapy programs, from assay development to validation and sample analysis. Our extensive assay list of over 800 validated assays is fully searchable online. We have state-of-the-art equipment, large capacity, broad capabilities, and LIMS to support both small and large sample sizes for non-GLP, GLP, and GCP compliant studies. QPS has a long history of technical expertise and began by supporting small molecule LC-MS analysis in the late 90s, expanding to ADCs, peptides, and elemental analysis in the early 2000s. Support of proteins such as large molecule biologics for PK and ADA by ELISA and RIA also began early in the 2000s. In parallel, biomarkers are supported with a variety of modalities such as ELISA, cell-based assays, LC-MS, and ICP-MS. In the next decade, more complex biotherapeutics such as ADCs and multivalent biologics required advanced technologies such as hybrid LC-MS, multiplexing by MSD, and automated nanoliter scale immunoassays using GyroLab platforms. Support of oligo-PK began as early as 2003 by LC-MS, with expansion by ligand binding and LC methods starting 2007 and 2009, respectively. Quantitative PCR services for genotyping, pyrosequencing, and relative gene expression by qPCR began in 2006. QPS gene therapy services expanded to support PK and ADA for mRNA and vectors in 2014 and 16, respectively. In addition, higher selectivity has been achieved for oligo-PK using LC high-res MS. More recently, QPS has added COVID-19 viral RNA qPCR and IgG serology tests. Here is a selection of approved drugs on the market that QPS has supported from pre-IND to post-market surveillance. Small molecule bioanalysis by LC-MS in Delaware has a team of over 70 scientists with over 25 years of small molecule experience. They have validated over 600 assays and analyzed between 200,000 and 300,000 study samples per year. This is a snapshot of the studies that QPS Delaware Translational Medicine has supported, broken down by preclinical rodent species, preclinical non-rodent species, and human clinical trials. The color-coded dots represent a single PK, ADA, biomarker, gene sequencing, or protein MS study based on sample number. As you can see, we have supported from small to tens of thousands of samples per study in support of a broad variety of study designs. For biotherapeutics, TLM has conducted over 150 drug programs in all phases on a variety of ligand binding platforms, and work on roughly 250 regulated PK studies annually, which includes gene therapy. For antibody drug conjugate programs, QPS has conducted over 25 ADC programs from pre-IND to phase 4. QPS's expertise in immunogenicity testing covers screening, confirmatory, and titer analysis, and we have completed more than 50 ADA drug programs in all phases, with roughly 200 regulated ADA studies annually, which also includes gene therapy programs. We have a lab dedicated to cell-based NAb bioanalytical services with over 12 years of experience. Since 2002, QPS has completed over 50 NAb studies for 7 sponsors based on ELISA, cell viability assays, activity assays, or receptor binding assays. Gene therapy is a fast-growing therapeutic area where QPS has supported the development of 45 oligo programs and 20 gene therapy programs from preclinical to clinical development using real-time qPCR, ligand binding, and LC-MS methods to support PK, ADA, biomarkers, biodistribution, expression, and comprehensive ADME. The work is conducted in a 5-room suite with directed workflows and separate reagent, oligonucleotide, and sample containment to prevent contamination. In closing, QPS is a global CRO with clinical and bioanalysis sites in North America, Europe, and Asia, with the global headquarters located in Newark, Delaware. We look forward to the opportunity to discuss your bioanalysis needs for current and future programs in your pipeline. Thank you for your time and attention.